while the forever young fish, none of the knockout fish were ever going into maturation and have this small spermatogonia. So just to end my seminar, I just wanted to sum up the gold standard for gene editing in fish, which relates then to disease, reproductive phenotype, sustainable field, and welfare and production phenotypes, and also then to cell line work, and also maybe to make cell lines which you can actually take into the fish to produce more of different genotypes. And the tools we're working is then embryonic injection outside the fish, cell lines, embryonic stem cell, surrogate production, and shortened generation time is also relevant because we want to breed in a shorter time frame than 3 years. And we also then interesting to see if we make edit, how that can be put into breeding strategies. So in the end, I just want to acknowledge my group at the Institute of Marine Research, which has done a lot of this work together with the Research Council of Norway, also EU project, and then a collaborator at Uppsala University and the and University of Bergen. Thank you.
Impressive. Thank you. Kasia, do you have questions?
Yes, thank you very much for the presentation. And there is one question coming from the audience, Michel Thibier. He writes that being not an expert in aquaculture, but what is the interest of albinose fishes? So
sorry, I forgot to say that. No, it's just a way to see that to reduce the number of animals we use. So in the beginning was just to show that we could get the CRISPR to work, but now we use it all the time. So whenever we are targeting one gene for a phenotype, we also target the albino gene. So then we only have to use the albino embryos in the screen for mutation phenotype. You know, because we usually get about 20% fish that has a very, very high mutation rate. And we can easily see those fish because they're yellow then. So instead of screening 1,000 fish, we're screening 200.
Yes, thank you for answering that. And there is the second question coming, which is the same which I wish to ask you too. So what is the successful rate of editing for the 10K per week?
For?
What is the successful rate of editing? I mean, could you, if it was not stated, the rate of successful injections and of successful editing?
Yeah, so if we have a good guide, obviously the albino guide is very good. The dead end guide we use is very good. We usually, so it's in the salmon, they have a very big egg, which a very little sort of cell on top. So whenever we hit the cell, we have a good success. So usually we have around 20% of the fish that we inject that has a high mutation rate. And that usually is right between 80 and 100%.
Yes.
So it's very, very efficient.
2,000. Yes. Mm-hmm.
So usually if we inject 2,000 fish, the aim is to have 400 fish of that type that we work with.
And another question coming from the first person from the audience: what pathogen would be first candidates for disease?
Yeah, so one of the biggest challenges in aquaculture is a parasite called salmon lice, which we are not working with because it's quite difficult. It's a parasite, but the first sort of aim we're working with is it's viral resistant. So now we have this project is working on a PD, which is SARS virus, and also IPN virus, which they know the target already. We haven't worked with that, but it's recently published the exact target of that receptor of that virus. But of course, in a dream situation, it would be to find a resistant to this parasite, which is causing the whole industry to sort of work, have to change all the production pattern just because of this parasite.
Thank you. Thank you very much, Anna. Now there are no more questions coming from the. Oh yes, there is one from Chris Schroten. What are current European rules with regard to the use of salmon obtained from gene editing?
Well, the current legislation is that they go through the GMO regulatory system. So they have to go through, like, you compare aqua bounty, which is the most known fish, it has to go through the same pathway, which is, if it was to be in Norway, we would have to send an application to Norway and to the EU, and then they would go in parallel.
You made a difference between loss of function and gain of function. Is either of them more easy than the other?
It's much easier to do loss of function. So we are working now with gain of, because we have been trying for several years to put in whole genes, which then is easier. But since it's CRISPR, it's not so easy to put in a whole gene. We're now changing the promoter and also the UTR of the gene to increase both the onset of the gene or the stability of the mRNA. So we are continuously trying to develop protocol to put in whole gene, but it's very, very difficult. And since the generation time is so long, even if we get integration of one gene, we have to wait forever. And then the fecundity is like 8,000 eggs. And then if you have 1% success, you have to screen 8,000 eggs to find 8 fish. So it's very, very challenging with the whole putting in a whole gene so far.
Because the fish is purebred production, I think, isn't it? It's the fish. You don't do crossbreeding.
I mean, the breeders have between different species.
Well, yes. Species lines.
Yeah, they do for. We have also been doing that. You can't create sterility by crossing salmon to, I don't know if it was rainbow trout, but there is possible to make hybrid fish, and then they are sterile. And it's done especially in, I'm saying it wrong, maybe carp. I'm not 100%, but it's possible.
Okay, thank you.
Thank you.
So yes, with this, the questions stopped falling for the moment, but I'm sure the question and answer box is still open. So please, audience, if you will have more questions, you are welcome to type them. And I think the next, our second speaker is ready for the speech. So please, the floor is yours.
So you can hear me and see all my slides?
Yes.
Okay. So first I would like to thank the organizers for inviting me, giving a talk on why and how to apply gene editing in pigs. I would like first to give you a short introduction of the technology, which describes the how, and then show you some application examples, which is the why. So as Anna already mentioned, we use the CRISPR-Cas system or genome editing tools in the same way as it is used in all the species. So with this technology, you can introduce targeted double-stranded breaks at any site in the genome, which causes this double-stranded break and triggers inner cell repair mechanisms. And about 90% of the cases, the double-stranded break is repaired by a repair mechanism called non-homologous end joining, which is a very error-prone system and can lead to the insertion or deletion of nucleotides, which then can lead to a reading frame shift, which results in a functional knockout of a gene. But we also use 2 CRISPR molecules, for example, to excise some proportion of the genome, which could be only a few nucleotides, or could also be some exons, whole genes, or even chromosomes you can delete by using 2 CRISPR molecules. And if you want to introduce some new sequences, like a SNP that encodes for a specific trait, you're relying on the so-called homology-directed repair mechanism, which is only active during G2 and the S phase of the cell cycle, and usually uses a sister chromatid to repair the damage in the DNA. But when you offer a template that encodes or contains your sequence that you want to add to the genome, you have some certain chance that it is integrated at the right side of the genome, mainly then gives a gain of function. So if you want to produce living offspring in livestock, we are relying on 2 technologies. The first one is zygote manipulation, and the other one is the cloning technology or somatic cell nuclear transfer. So when you do zygote manipulation, you can use in vitro or in vivo derived zygotes, and then micro-inject your CRISPR-Cas molecules into the cytoplasm of the zygotes, or you can electroporate the zygotes and use that way to bring your CRISPR-Cas molecules into the zygote. And then you can transfer the early embryos into recipient animals. With the cloning technology, you use primary somatic cell from an individual, do the gene edit in the cells. You can screen the cells, select for the best cell clone, which contains your gene edit, and then use these cells as donor cells in the nuclear transfer procedure, where you replace the genome or the sequence that is in the oocyte is then replaced by the somatic cell genome. And then you can transfer these embryos to recipients, and you get an offspring with the desired gene edit. So these are the 2 technologies that can be used in livestock to produce edited livestock. And there are 3 sectors in which gene editing is mainly used, especially in pigs. So the first sector is agriculture, where we try to address animal welfare issues, like, for example, in reproduction, where we tested if we can use genome editing to produce animals with a specific sex, or where you can use them as surrogate sires, or you produce animals that are disease resistant, for example, against severe diseases like African swine fever or PRSF or classical swine fever. But there are also applications for increasing the production, to increase the growth or development of the pigs, that they have a higher meat production, or that they are more environmentally friendly. Or you can also make the energy homeostasis working better, so that they produce more brown fat tissue, and you can also use it for dietetic improvements. And genome editing has also extensively and intensively promoted the application of pigs as human disease models. So changing the genome allows these animals to resemble the mutations causing genetic disorders in humans, and pigs phenocopy human disease manifestations more accurately than the commonly used mouse models. So here are some disease models like cancer. There have been pigs produced that have a Duchenne phenotype, or Alzheimer and Parkinson, and etc. And pigs are also considered to be an optimal donor, organ donor for xenotransplantation. And of course, pigs are also used for basic research, like to find out to answer some immunological questions, or to find out what the specific function of a gene is, for example. So, and I would now like to come to the why we apply genome editing. And one example I want to show here comes from our laboratory, that is to use genome editing for sexing. And in the pig, you have this specific problem that causes some animal welfare issue, that is boar taint. That means that the male pigs produce a specific odor which is perceived during the cooking, and about 75% of consumers are sensitive to boar taint, which is described as an intense fecal and urine odor, which is produced by the breakdown product of tryptophan and androstenone. And especially women in certain ethnic groups show a higher degree of sensitivity. And due to the boar taint, routinely practice is that these piglets younger than day 7 after birth are castrated without any anesthesia. And that is done in many parts of the world. About 75% of all male pigs are castrated without anesthesia. And this causes animal welfare issues because these piglets suffer from pain and stress, and they also have a higher risk of infections. So castration without anesthesia is prohibited in Germany since this year. And there was a long discussion on alternatives like castration with anesthesia, like boar fattening and immunocastration, but only boar fattening leaves the pigs completely untouched. But it has also some other contrasts like restlessness and dominant male pigs, penile injuries occur, and you have to detect boar taint, which is necessary for the consumers so that they don't get meat that has a boar taint. So you see there are many pros and cons regarding the alternatives, and we try to find out if we can use genome editing to address this issue. And it is known, or it was known from the mouse, that is specific expression of a transcription factor which is called SRY, and which is encoded on the Y chromosome, is crucial for the sexual development. So if you have expression of this transcription factor from the Y chromosome, it triggers the expression of other genes like SOX9 and FGF9, which then leads to a testicular development. So that means this offspring has a male phenotype. If you do not have expression of the SOY gene, other genes are becoming activated and you see ovarian development. That means a female offspring develops. So we wanted to see if that system works also the same way in the pig. And we used the CRISPR-Cas system to excise a huge proportion of the SRY gene from the porcine genome. And we injected these 2 CRISPR molecules into in vitro derived zygotes and transferred these embryos back to 3 recipients, of which 2 became pregnant. And they delivered in total 12 offspring, and all of them showed a female phenotype. So we did not use any sex sperm for the fertilization. So usually we would expect a 50 to 50 sex ratio, but we only got female offspring, phenotypically females. And so we checked the offspring, and in 3 of them, we could clearly detect a Y chromosome. So these 3 offsprings were genetically males, but they showed a female phenotype. So, and also the inner genitalia were almost the same like females. So they developed a uterus and they developed ovaries. It became obvious that these ovaries are a little bit smaller than the ones from female offspring. And this difference became more obvious when these pigs got older. So this is a picture of 9-month-old pigs. And you can clearly see that the vulva is much smaller compared to wild-type controls. And the uterus is immature, and you do not see any functional follicles on the surface of the ovaries. So these SRY knockout pigs, they did not show any signs of estrus, so they were infertile. And in the next project, we are trying now to find out what other factors are necessary for the maturation of the uterus and also for the ovaries. So another project is to produce disease-resistant pigs, and African swine fever is spreading all over the world, which is endemic in Africa, as the name says. And natural hosts are the warthog and bush pigs, which are resilient to infections, and it's transmitted directly or via ticks. And our domestic pigs are completely susceptible to this virus and develop an acute and rapidly fatal hemorrhagic fever, and die within about a week. So there's no vaccine so far available. There's some progress regarding the development of a vaccine, but there's no vaccine so far available for commercial use. It has a very high mortality, up to 100% of the pigs die. So the only strategy to prevent spreading of the disease is culling of infected pigs. So far, official losses are more than 8 million pigs, but it is estimated that China has already culled about 150 million pigs. So these are pictures that consumers and also veterinarians and other people don't want to see, all of these huge amount of culled pigs. So we started one project where we integrated a CRISPR-Cas vector into the genome of pig cells that express the Cas9 and also guide RNAs that should target the genome of the African swine fever virus. It targets a specific gene which is important for replication of this virus. We tested the cells in vitro studies. So these were wild boar lung cells that were transfected with our construct, and they showed resistance against infections with the laboratory strain of African swine fever and with the field strain, Armenia, but they were susceptible to infections with the Kenya strain. And when we sequenced the Kenya strain, we observed 4 mismatches in the sequence that we targeted in the gene. So this was some sort of rescue mutation for this viral strain, but it showed quite good that the CRISPR-Cas expression led to a 10,000-fold reduction of viral replication, which should also provide protection in vivo. So we have produced pigs that have integrated this transgene in the genome. We started with initial in vivo experiments that were not that successful at the beginning, but we have some idea to improve the whole system. Another much more successful approach was the generation of Prs resistant pigs from 2 groups in the US and in Roslin in Scotland. So PRSS is the porcine reproductive and respiratory syndrome, and it has been one of the most economically significant swine diseases worldwide for over 2 decades and has been estimated to cost the US swine industry at least $600 million annually, and the annual cost for the EU reach 1.5 billion euros each year. So it's a very important pig disease. And the receptor for that virus is known, it's CD163, which is mainly involved in hemoglobin degradation and inflammation. So what is also known is that the virus does not bind to the whole receptor, but only to the 5th chain of this receptor. And the group in Roslin, they used a very sophisticated approach and cut out the exon 7 of the CD163 gene, which encodes for this 5th element. And they maintained the reading frame so that the physiological properties of the CD163 gene still remained intact. So these pigs were completely protected against infections with different strains of PRSV. So this is a very important model and shows the strength and the capability of genome editing in pigs. So a biomedical application is xenotransplantation. We have also a huge project dealing with xenotransplantation. And what you can see here is the active waiting list for a kidney transplant in Germany. And you can clearly see that there is a huge number of people on the waiting list and only a few available organs. So there's a huge gap. And that leads to the fact that every day 3 patients in Germany die while they are on the waiting list for an organ transplant. So the average waiting time for a kidney transplant in the EU is 5 to 6 years. And this is a worldwide problem. So there's a huge organ shortage worldwide, leading to the death of many people that are on the waiting list. And the pig is now considered to be the perfect organ donor because it can be kept under highly hygienic conditions. The genome reveals high similarity to the human genome. Also, physiological parameters are quite similar to humans. The organ size matches quite well, and there are less ethical concerns. You can easily propagate these pigs, and also the transmission of pathogens is considered to be of minimal risk. But there's one severe problem. These are severe rejection response, which would occur when you transplant a native pig organ, because the pigs have specific sugar molecules, which are called gall epitopes on the cell surface, which we lost during evolution. And about 1 to 5% of all circulating antibodies in your blood flow are directed against these gall sugars. So if you would transplant an organ, a native pig organ, these antibodies would bind to the gall sugars and would trigger the rejection cascade, leading to a hyperacute rejection of this organ within minutes. So what we know so far is that we have to do extensive genome editing in the pig genome to make pig organs suitable for xenotransplantation. And this includes the removal of the gall sugars, but also removal of Neo5-GC, which is another sugar molecule, and the products of B4-gality2, which is also some carbohydrate on the cell surface of pig cells. And we also need expression of human genes to overcome the incompatibilities between the porcine and the human coagulation system and also inflammation system. So with the genome editing technology, we can achieve all these goals. And what you can see here is the so far most intensively modified pig described so far for xenotransplantation. So these are pigs that have a triple knockout or even a quadruple knockout with a perf knockout, and it expresses 9 human transgenes. So these were generated by an American startup company, eGenesis, which is located in Boston. So the maximum survival time of a pig organ after transplantation into baboons, which is the preclinical model for xenotransplantation, dramatically increased after genome editing became available. So when you transplant a pig heart into a baboon, orthotopically, that means that you remove the baboon heart and it is replaced by the pig heart. We have now the longest survival, which was recently described, of 264 days, which matches the requirements of the FDA and the WHO for getting into clinical trials with xenotransplantation. So it is now anticipated that clinical trials will start within the next 5 years. So pig production is facing many challenges like climate change and the reduction of greenhouse gas emissions, a growing world population and its demand for food supply, also animal welfare issues that have to be addressed, and also prevention of disease outbreaks. So in all these challenges, I think can be addressed not alone, but with genome editing that could help and could be one piece of the puzzle to address all of these challenges, because it is a very efficient way to integrate or to propagate specific genetic traits into the livestock population. So as I showed you, it can be used to render livestock resistant to severe pathogens and to address urgent questions in the animal welfare context. So from my perspective, it could be an essential part of future breeding strategy to also integrate specific traits into the breeding herds rapidly. So, and it can be used to establish new human disease models in the pig to develop new therapies. And gene-edited pigs can be the solution for the urgent demand of suitable organs for xenotransplantation. And with that, I would like to thank all our collaborators and especially all the members of our lab, and especially my very dedicated and talented PhD students that work on the projects that I have shown you here today. And yeah, I would be happy to answer your questions. Thank you very much.
Thank you for being so nicely in time and so stimulating and motivating. Well, one of the questions we had is the academia. What type of students should they start training looking at this future? It could be the gene editing itself, or it could be the reproductive side of it to create more efficiency, or it could be the management of animals. I can imagine that these xenotransplanted animals need proper care. So where should academia go?
Well, I mean, it's, I mean, we have a good, I mean, we have a lot of people that are coming from different areas. We have veterinarians here. We have people that come from agricultural universities. So I think it's a good mixture to have everything together, and also people from human medicine, because I mean, they give the input what kind of human disease model would be interesting for them, where they're still lacking an interesting model. To bring it into commercial use, I mean, of course, it would be good to have more people from agricultural universities that get aware of this technology and see the benefit of this technology, especially for breeding purposes.
Thank you. Kasia, do you have questions? Because we have more questions.
Yes. Yes, thank you for for that answer too. I'm also interested in that answer particularly, but the questions from the audience. One of the listeners did not catch about the African swine fever on the Kenya strain. Is it understood correctly that on which gene targeted did you get a 10,000 reduction, 10,000-fold reduction?
That was on the
viral load, I think.
Yeah, sorry, maybe a little bit too fast, but I mean, that was in the laboratory strain and in the Armenia strain. Armenia is also a field strain that is, as the name says, is in the Caucasus, very active. But they were not protected against the Kenya strain, because the Kenya strain has 4 mismatches in the target sequence. And this was some sort of escape mutation. But we could easily adjust that. So we have designed now specific guides for the Kenya strain as well. So we can also use a multiplex vector that transcribes several guide RNAs for the specific strains.
Thank you. And keeping on in that area, is there progress for resistance of pigs to the other African swine fever strains, which you probably now answered?
Yes.
So, and one of the persons from the audience asks why you don't inactivate the androstenone pathway and one of the enzyme, for example.
I mean, this is done by another group. Which is it's called it's a company, Recombinetics, and their daughter company Xenotransplantation. So they are working on producing pigs that have a knockout of the kisspeptin, which is belonging to that pathway, and these pigs do not reach puberty. So this is another approach. We use that approach because we did not only address the animal welfare issue regarding castration, but it's also a good model for human disease, because this is also occurring in humans, that they have a mutation in the SOY gene, and they come to world as a baby girl and are treated like women, and then they do not reach puberty and cannot or do not get pregnant. And at the end, they find out that they are genetically males and not females. So this is also some sort of human disease model. So so we have two sides of the coin where we can use these pigs for.
Yes, yes. So for your research, this is another question. Does the current EU regulation pose any problem for you?
Not for research.
Research.
I mean, for research, it does not play any role, because I mean, we are belonging to the German government, so we belong to the Federal Ministry of Agriculture and Consumer Protection, and not consumer protection, we have agricultural Ministry of Agriculture, and we do. I mean, we do the experiments to give informations to the politicians what can be done, how this system works, what are the pros and cons, and where they maybe have some information which make a basis for their decisions. So this is it's important what we do, and the legislation does not play any role for research. It's important for commercial applications of genome editing, and this is it's a drawback, the legislation so far.
So if I just keeping still to this topic, is if I understand well, you do not need to obtain special permissions or specially structured laboratories, or do you? Because there are some regulations that you, for example, you need to have special governmental permissions in some European regions to conduct the transgenesis and research on GMO, and then to raise the animals. So is this also an issue for the gene editing research?
Well, sure. I mean, we have some restrictions, so that, but they're not that huge, because they are just kept under what we call security level 1 conditions. So that you have a containment, that these animals cannot escape, but these are just very low restrictions. So it's okay.
Okay.
And I see these questions from Michael Tibier, so I did not understand his question. The gene, okay. Well, the gene was something with, well, I have to find, I can send him that by email, or I just type it in the answer for that gene. But you can easily read it in our publication.
Well, thank you. So that will be very satisfactory. And the one question just above is about the, so could genome editing make old breeds working well on low input farms, resistant against AFC and PRS without changing them in another way?
I mean, that is a great benefit of genome editing, that you can add a specific trait into the genome and leave the rest of the genome untouched. I mean, that is a great benefit. And that is also, I mean, also vice versa, the system works. We are also containing the German gene bank. So we have stored here samples from old breeds of poultry and cattle and pigs, and it would be interesting for the future to sequence the whole genomes of these animals and to find maybe some new traits which might be important to address challenges of the future, which we could then use and integrate them into our breeding lines, for example. And you leave all the other things untouched, and that is a great benefit of that technology.
Yes. So if, yes, we still have time. The last question here is about the safety concerns regarding the endogenous retroviruses in pigs. Is this a concern still if used as transplant donors?
Well, it's some sort of concern, but only of a very, well, it has really decreased within the years, because we had, initially there was one publication describing that porcine endogenous retroviruses can infect human cells and maybe could recombine with human endogenous retroviruses. And it was shown that it's only the Prs that can recombine. And it's now, we screen our pigs that they are Prs free, and you can now easily, but you can knock out Prs in the or Perf in the porcine genome. So that was done by eGenesis, for example. They knocked out all endogenous Perf in the genome, and then these pigs are safe. And also from the authorities, Perf is not considered a real threat for xenotransplantation, even without genome editing.
Thank you. Hopefully that really answers the last question. I would have one question regarding the in vitro models and well, developing the techniques for transfection in vitro, because one of the techniques used for gene editing is cloning, through cloning. So is there a sort of preferred cells, somatic cells that you use for that, or you use fibroblasts as you showed in the presentation, or there are other also type of cells that you could use?
I mean, yes, we usually use fibroblasts because they are easy to culture. They have some good lifespan and so on. We can use fibroblasts from different origins, like maybe fibroblasts from kidney cells and so on. So they have a longer lifespan, for example. But that's our preferred primary cell. But other groups can use also mesenchymal stem cells. I mean, it was shown that you can almost use any primary cell for that, but if you want to do gene editing, it's good to have a long lifespan to screen the cells and that they are still usable for somatic cell nuclear transfer.
So do you also find, well, continuing on this, do you find some, how would you call it? I mean, do you find some preferable techniques of transfection? Because electroporation is one of them to deliver the genes, yes, and the, well, CRISPR-Cas, the editing tools, but do you also try another techniques other than electroporation, or use electroporation as a standard for?
I mean, for the cells, we use electroporation because it is the most efficient way to get these CRISPR-Cas into the cells, in our way, in our laboratory so far. For the zygotes, we still use the microinjection technology, but we are also trying to establish a protocol for the electroporation for zygotes, which seems to be also quite efficient. We tried also other ways like lipofectamine and all that stuff, but I think the electroporation, especially with the new devices, is very efficient.
Yes, and efficient, could you share what rate do you consider efficient for your research? Well, at which rate you are satisfied to continue?
It depends what we want to do. I mean, our best, usually when we knock out a gene, we do not use a single guide. We use always two guides to delete a certain proportion of the gene because it's much easier then to detect and you know what you deleted. So when you just use a single guide, so you don't know what really happens. Sometimes the reading frame still maintains okay, and then you just have no functional knockout. So we usually delete a certain proportion. And I mean, when we get efficiency up to 30 to 90%, I mean, that's okay, because in the primary cells, we can screen for the right cells. So efficiency is not that important. It's more important for HDR. And there we get very low efficiency, only about 2% of the cells. So it would be good to achieve maybe 10% of the cells.
Yes, yes, but that's important. That's the technical issue. Thank you very much, because I think that this is really thank you. And Egbert, if I am right, we need to invite all of you to the coffee break now.
Yes,
that's
please.
Right. So go to grab your coffees, and I think we meet in 10 minutes. Yes.
Yep. Thank
Good.
you.
Thank you.
I'm here.
Hello, hello, Mike.
Hello.
Good to see you. You haven't changed much since yesterday.
Well, I'm using my home computer at work, so I hope it works. I've never given a talk on this, so I hope it goes well.
It will be very fine.
Well, let's hope.
And the other two speakers did a tremendous job, so we have very high expectations from you.
Oh, thank you. I always, when I give a seminar, I always, if the person before me doesn't give a good talk, I always feel ready to go then. And now both Anna and Björn gave, really excellent talks. I hope they're listening to me when I said that. Yeah, that was very nice talks.
They could raise their hand or applaud.
Yeah,
there we go.
All right, do. Go, Mike. You always do a tremendous job.
Our idea is to have some direct question to Mike, and then afterwards have all the Björn and Anna also on screen, that we have screen with the pictures, and then do the general discussion.
So Egbert, you can invite Mike to the floor.
Oh, Mike, you're invited to start your presentation.
Oh, thank you. Is that it? Straight away. All right. Okay. How is that?
Right. It's good.
You don't see the big banner at the top, do you?
Now we see you and the presentation, and
very
you
good.
Can,
okay.
Proceed.
Thank you. Thank you. I've had problems with this before. So I'd like to thank you for the invitation to speak today. It's really an honor. And I'd have to say, I really enjoyed the first two talks, talking about fish and pigs and the genome technology and how it's used in those species. So what I'm going to talk to you about today is our targets for genome editing in poultry. And you see I have a question mark there because I do believe that we don't have as good of targets as we have in these other species. But at least I hope you get the idea from my talk is that we can use these tools to actually really investigate gene function in chicken. So first I'm going to start off and I'm going to talk about the technology for making changes of DNA in an organism. And you've already had two really nice introductions, and how we make those changes in a bird species, and also how we're using this technology going forward, because there's some really interesting things we can do in birds. And I'm going to touch let me start my timer. I'm going to touch just a little bit about the current regulations involved in using genome editing, because it changes per country and it changes, it's in rapid flux, especially here in the UK. So a brief introduction. So you could imagine where all animals are made of, we have DNA, which is, you could imagine, is our software for encoding for us, and DNA spells out letters, and you can imagine these as letters of in a sentence, and the sentences come together to make paragraphs and chapters. And when you put all this code together, you actually encode all the information you need to make that animal. So that's why DNA is so important for us. And each animal has, each bird has a slightly different code that allows it to be put together in a specific way. So changing and altering this code actually tells us a lot about how life and how these genes work in this animal. So changing the genome has traditionally relied on evolutionary chance mutations. So a mutation can occur, and then the farmer will observe that animal has a different size or grows faster, and it's selected for. In that way, that's selective breeding. But also in plants for the past 50 or 60 years, mutagens have been used, and these are chemical and also irradiation of plants, and they would go in randomly and change a few base pairs or even larger in that plant species. And then the farmer could go back and select select that animal or that plant to be brought forward and put into production. And this actually, over the past 80 years, a huge number of plant species have been generated in this way. There's over 3,200 varieties that have been produced through either irradiation or through chemically, and their progeny. So this is led to large modifications of plant species that have gone into food production. And these animals or these plants are not regulated as GMO. They're just, they're actually not regulated at all. And this is in most countries in the world. So jumping from this random mutation of DNA forward, we have this new tools that you've just heard about over the past hour, and these are called genome editors. And what's amazing about these is that rather than, depending on nature to go through and create a mutation in a particular spot in the genome, we now have the tools that we can precisely go into this book of life, and change a single letter on a single page in that book. And that's what's so exciting about this technology. There are 3 different kinds currently of genome editors that we use. These are called, 20 years ago, we were using what was called zinc finger nucleases, and these were repetitive protein that recognize a specific site in the genome. These were followed up by what were called talons, which first emerged about 10 years ago. Again, this is protein, and you put different domains of these proteins together, and each domain would specifically recognize a different letter of DNA. So when you put these together, you could recognize any site in the DNA, and these would be fused to a restriction nuclease, which would cut the DNA at that point. And now, as you probably know, we have our new tool, which is called CRISPR-Cas9. And this relies on a protein and an RNA antisense molecule, which will recognize any site in the genome and cut it specifically. So all these tools have in common is that they're site-specific nucleases, and they would go in, if you add in this case, Cas9, it will go in and cleave a specific spot in the genome. And what happens there is a process called non-homologous end joining, which the cell will try to repair this break in the DNA. And this can lead to something. Sometimes the cell will repair it perfectly using the other chromosome as a copy, but many times it ends up in an error at that place, which would cause an insertion or a deletion. This can be very small in size. And this process of genome editors using NHEJ is regulatory known as a SDN1 change, and that'll come up at the later part of my talk. The second thing we can do is you can give a template, which would leave to homology directed repair. And basically, you cut the DNA at a specific spot, and then you can give an oligonucleotide to lead to the repair of that spot. And this repair can just change one base pair or a few base pairs, or it can actually change to several hundred to several thousands, and you can even insert a gene at this area. And this process in terms of regulatory is called SDN2 or SDN3 change. And SDN2 would be when you would just put in a few up to 10 base pair changes at the site of cleavage, and SDN3 is any change that would be larger than that. So I don't want to insult Björn by saying, I used to say genome editing in mammals is easy, but that's anything but true. Genome editing in mammals is becoming relatively straightforward for many, many livestock species. Always this depends on generation of a mutation. And then this is followed up by breeding. Which is also possible is that you can genome edit a cell to put your change into that cell, and then you can use that cell for cloning and these would be inserted in this single cell zygote, and then the whole animal would develop from this. So birds have genome editing in birds, and genetic modification of birds has actually, or for avian species, has been very difficult. And this is due to the large size of basically the zygote here. And this is the laid chicken egg. This is showing it for chicken, which has this huge yolk, this is a single cell, and this is centimeters across. And you know this from, if you eat eggs, this is from your breakfast. And on top of this is actually a single cell. On top of this is a pool of cytoplasm. And in that cytoplasm, there's a single nucleus. So to deliver these genome editing tools is very difficult in bird species. And also I should mention that cryopreservation of avian species is also difficult because freezing down this structure is at this point impossible. So rather than genome editing this zygote, what many laboratories do, including mine, and this is for chicken, is that we actually edit the germ cells, or the primordial germ cells that you heard Anna talking about during her talk. And what we can do is these early germ cells, so all they do is go on to make the sperm or the eggs, but they're born very early in the chicken embryo. So we can simply go into this early embryo, and at this point of development, this is only day 2 of incubation of the chicken egg, and it'll hatch at 21 days. We can simply go in with a little capillary pipette and take out a microliter of blood. And in this blood are a few of these primordial or young germ cells. So their purpose in life is to migrate to the gonad and to form sperm and males and oocytes and females. So here's us sampling this. We can then take this little bit of blood, and we're actually able to culture these primordial germ cells. We came up with a culture medium that is defined, and we can simply take these few germ cells and put them into culture, and they will grow over a month and make dividing once every 24 hours and making over 100,000 cells within a month. So at this point, we can go in with our genome editing tools, CRISPR or Cas9, transfect the cells, and then we can also grow these cells out clonally. So we're able to select for the genetic change that we will want in these primordial germ cells. In this case, we put in a gene just that encodes for green fluorescent protein. So at another point, at a later point, we can freeze these cells, or we can select for them, and then we would inject them back into a host embryo, same stage. And then simply what we can do is we can cut a hole in the shell, inject them into the vascular system of this early embryo. It's very welfare friendly. We seal the embryo up with a piece of tape and we can hatch it. So that's our surrogate host carrying these genome edited PGCs. And then if we raise it and mate it, in this case, we put in a GFP gene. So some of the offspring came from that surrogate, they came from the surrogate host, but they came from that donor germ cell that was genetically modified. So this is our platform for actually introducing genome edits into a chicken. This process, I should say, is very inefficient because the germ cells that we transplant in have to compete with the endogenous germ cell. So surprise, surprise, we actually took the approach that Anna told you about of making sterile surrogate hosts. So the idea was if we could engineer sterility into the surrogate host embryo, then when we injected our germ cells, they would completely repopulate the host. And then when we raised it up, all the germ cells in that animal would come from our donor germ cells. So in this case, we injected male germ cells. We could inject them back, and they would repopulate the male testes If we use female germ cells and we put them in a sterile surrogate host, they would repopulate the female oocyte. And the idea we had is actually we could directly breed our surrogate hosts together. So we could breed our sterile male to our sterile female, both carrying the donor germ cells that were genome edited. So in a single generation, 100% of the offspring would come from our genome edited cells. So we could use this for making genome edited chicken, or we could also use this technique for biobanking. And that's what I'm showing you on this slide here. So this is basically, I haven't given you a lot of the details of the technology of how we make the sterile host, but it's through a transgene. And basically what we can do is we can take any breed of chicken, we can sample the early embryo, and we culture the primordial germ cells from that breed of chicken. And I should mention it's only for chicken that we can do this now. At this point, we can freeze down these cells and we can store them. We can then genome edit them in culture. We can put in whatever edit we want. And then we can introduce them back into an egg of our surrogate host, where if we mix it with a compound that induces a transgene, it will delete the endogenous germ cells. So we can then raise up these surrogate hosts and breed them together, and they will produce pure offspring. So basically, this is our whole platform. And I should say, this was probably 7 years of, or even 8 years of work to put this whole thing together. And this is showing, this actually captures how we're using this technology. We're using it to biobank by freezing down germ cells from different bird species. And I hopefully this captures the power of this technology. So in the back here, we have a breed of chickens. These are the layer breed, and these are our surrogate hosts. So they're these beautiful brown chickens, and they carry the gene for induced sterility. And we transplanted germ cells into these surrogate hosts from a different breed of chicken. In this case, they have a black skin, so they weren't genome edited at all. They just happened to be a silky breed, and this is broiler. We injected them into the eggs, we bred, these are the parents, and these are the offspring. So this shows how we can biobank and also genome edit different breeds of chicken. Oops. So we're using this technology. I'm just going to touch on a few things. So the chicken actually is a really good model for studying developmental biology. And as I saw, it's because the embryo, you can get the embryo without having to kill the mother, which is something you can't do with mammals. So using chicken embryos is welfare friendly. It addresses the 3 Rs, and we can study a lot of the same processes that occur in the chicken embryo occur in the mammalian embryo. So we've made different fluorescent chickens using different markers to identify different tissues to study the early development. And using our sterile surrogate host, it actually offers us a platform to be able to do this. So now we can just tag male germ cells, and we can put in our GFP reporter in those male germ cells, and then we can inject them back into the sterile host. All we have to do then to generate our transgenic line is to breed from this male to wild type female. So this is the whole process. We only have to make one sterile male carrying our edited germ cells to produce fluorescent embryos for our experiment. And as I said, this is showing you one example. You simply can cut a hole into the egg here, and you can observe the embryo. You can put a bead in, you can do a manipulation. And this is before the nervous system has formed. So this is really the power of the chicken embryo. And on the right here, I'm showing you, this is experiment by Michelle O. And we've used PGCs that were edited to contain a GFP reporter. And we bred that to a wild type to produce these, wild type hen to produce these eggs. And we can simply watch this. One of the things we can watch is development over several days just within the egg, and it develops completely normal. So this is going from day 4 of incubation up to about day 7 here. So this is the early stages, and we can study the whole process. So that's really one of the main uses we can use for this technology, is really to study the development of the chicken. So genome editing, as I said, is a very powerful tool. So we wanted to carry out a proof of concept, and we decided to edit its production, well, you could call it a production trait, but many of the commercial breeds of chicken have white feathers because, and this is for white leghorn, and this is because they contain a mutation in a gene that's called dominant white. So what it is a 9 base pair insertion into a gene called PMEL17, and PMEL17 is important in melanocytes to make pigment. And when it's mutated, the melanocytes can no longer make pigment. So it's a natural mutation that's found in commercial chicken breeds. And what it is showing the locus. What it is it's 3 amino acids, W, A, and P, or I should say tryptophan, alanine, and proline that is introduced in frame into the genome of these chickens. So we decided to actually take this breed of chicken and remove this mutation to see if we could actually restore the color to these birds. And this is, I should say, this is work from my postdoc, Maeve Ballantyne. So she went in with a CRISPR-Cas9 and 120 base pair template that contained a deletion in this locus. And this is showing the sequence here of a wild type, again, taking the germ cells from this breed of chicken containing the dominant white. And this is after she edited out this mutation. So she did this in both male and female germ cells. So we had them. And then as the proof of concept, we injected them back into our sterile surrogate host, and we bred the sterile surrogate host together. And this shows the result. So this is Maeve here who did this work. So again, the offspring, we bred the line of chicken we bred was called line 6. It's an inbred line of 6. It's an inbred line of chickens, and it's actually not very fertile. And this shows the normal line 6 offspring, or containing this dominant white gene. And on the left here, it shows us this is the offspring from our surrogate host. They're pure line 6 chicken. And look, they have this beautiful black feather color. So we sequence these afterwards, and these they actually lack this mutation. So we remove this natural mutation that occurred in these chickens, and we reestablish feather color in these chickens in a single generation. So I've given you 2 examples of how we can use targeting. We can use genome edits in poultry, and supposedly 2 targets. One for chicken is a good model for developmental biology. So by using genome editing, we can actually really investigate genes and their role in early embryogenesis. 2, we can use genome editing for validation of beneficial alleles. So again, this is for the dominant white. So when first they found the dominant white mutation, they suggested that its role was to make pigment, but it was never tested in an animal because the laboratory that discovered it didn't have the tools. So now it means that if a mutation is discovered in chicken, we can go in and investigate it to see if it's actually a valuable beneficial allele for feather color or even for production traits. So in terms of to think going forward, if we would say what traits would be used or what targets would be used for genome editing of poultry in production, well, the #1 target that I would say, I think, would be welfare targets, and this would be for disease resistance. So as you know, for poultry, we have many pathogens that most can be controlled with vaccination, but several can't. #1, avian influenza is the killer. We don't really have good vaccines for avian influenza. And every year we have outbreaks. They seem to be occurring more often. So any way to genome edit to bring resistance or discover resistance into poultry would be very useful. Also, Newcastle disease is a very important disease, even though we have a really good vaccine for that. or a vaccination program for that. Still, there's some chickens that can't be vaccinated, so perhaps genome editing for resistant would be useful. And another RNA, another virus is avian leucosis virus. The third thing, as you heard about from our other speakers, is that perhaps we could genome edit for economic traits. But again, this is an animal. Should we actually be genome editing to make it more productive? But in our world now that that is vastly overpopulated and in need of more food production, perhaps without increasing greenhouse gases, perhaps genome editing for economic traits would be useful. So in the final few minutes here, I'm going to show you some work. One of the work, before I show that, first, I would like to point this out. So I said genome editing for economic traits. And I would just like to point out this talk by Bill Gates. So Bill Gates, actually from the Gates Foundation, funded our project to make, through the Center for Tropical Livestock Genetics and Health, they funded our project to genome edit the dominant white allele as a proof of principle. And I think Bill summed it up really well, because he said his goal was to help smallholder farmers, so subsistence farmers. So his idea was, well, what if we can vaccinate a farmer's animals so they don't die? Well, this is a very good goal. And then his thing he followed it up with is, well, we need a cold chain. So what if we can actually deliver these vaccines to places they're needed so we can vaccinate these animals? So getting out the vaccines to the smallholder farmers to be able to vaccinate their animals. And then he also posed this question saying, well, what if we can genome edit an animal so it no longer needs to be vaccinated to be disease resistant? So this would be very important for Newcastle disease virus, as in if the animal is already resistant to Newcastle disease, we wouldn't have to be able to vaccinate it. And I want to point out this paper. So this actually has been done, and this is by colleagues of mine in the Czech Republic, and they use CRISPR-Cas9 to edit the transmembrane protein called NHE1, and this is actually the receptor that was used by avian leucosis virus J subgroup to get into the chicken cells. So again, this is similar to what Björn showed us by editing the receptor for CD174, I believe it was, made the pigs resistant to Prs virus. They edited this receptor by changing a single amino acid that the pigs were now, the poultry was now, the chicken was now resistant to avian leucosis virus. So this was a very important piece of work that they did there. Unfortunately, commercially, they didn't have a market for this. So these genome edited chicken weren't brought forward, which is a big problem for this. The second thing, the biggest target, I would say, and I'll just finish up in the next minute here, is for genome editing for bird flu resistance. So as you know, bird flu is caused by influenza A. It causes severe disease in birds, and it can actually lead to zoonotic diseases or transfer to humans. So it's a very big concern. And I do have a project that is funded, again, by a commercial breeder, actually through funding by the BBSRC and a few of my colleagues. And we have a project to generate genome edited resistant chickens through changing a single amino acid in that bird. But that project is ongoing. I'm just going to sum up here. So I think genome editing is becoming relatively straightforward in livestock species. We can make precise base pair change, and the chicken is now very amenable to genome editing. Some plant projects that are produced by NHEJ are actually not being regulated as GM food, and in a few countries, Argentina, Brazil, and Canada. Of course, this excludes the EU. Well, and you probably know in the UK, we're looking at our gene regulatory tools now. So perhaps plants going forward would not be regulated as GMO the ones that were produced by NAGJ. For animals, of course, this isn't the case. You might have heard recently in the news that they knocked out the gene for myostatin, which controls muscle growth in sea bream, and the Japanese government said this would not be regulated as a GMO food. So again, in fish, this is a case of using NAGJ in a fish to lead to deletion. So this would be interesting because this doesn't increase the welfare or improve the welfare of the animal. However, it does lead to a greener animal because that fish would be produced without, you would get more muscle mass with less food input for that sea bream. Just there to mention the people who've done this work, and I mentioned the names earlier. And I'll stop there and stop sharing, and thank you for your attention. Thank you.
Thank you very much. It was a beautiful story on the how, and you started, I'm not sure about the what, but at the end there was quite a bit of what. You very nicely unshared your screen, so I would like to ask Björn and Anna to show themselves online and then have kind of a bit broader discussion. Because Anna started with the what, and my feeling was that the how in fish is easier than in poultry and in mammals. But after Mike's presentation, I start to doubt that the chicken platform seems to be quite efficient. So would the three of you that like to speculate a little bit if it's easier in one species or the other, gene editing?
I was going to say, I've spoken a lot. I'll let the other two speakers make a comment now.
Okay, Anna.
Well, thank you very much, both of you, for nice presentations. I think, I mean, for some cases, fish are easy because, we're outside the fish. When we're doing the editing, of course, it's easier. But we don't have, for the cultivation of germ cells, it's possible to do in fish, but it's more challenging. It's not so easy to cultivate fish cells. So that part, sort of making that they multiply in a cell line, is not that easy. And plus, we have the generation time, which is awfully long. I mean, so we wait and we wait, But then we have other things that's good. We have a high fecundity. So when we have a good edit, we can really upscale quite fast. So,
I must emphasize that PGC culture in chicken is not that easy as you might think from Mike's talk. So they are great in doing that. And I mean, in the pig, I mean, if you just want to achieve a simple knockout, it's quite efficiently to achieve by using electroporation of zygotes. You don't need much expertise in doing that, and then transfer these embryos back. Also in other livestock where you can do that and then transfer the embryos back, for example, in cattle or something like that, so that you're not relying on the cloning technology. And we have also some other cells that are becoming available. We have to work on the protocols which are potential or expanded potential stem cells now also for the pig and for cattle, which have almost the same, or they have even more capabilities than the embryonic stem cells in mice. So I mean, this is a technology which could maybe change the whole field in the future.
Mike?
Yeah, I was going to say one of the important things is the welfare aspects, and that's why I'm jealous of the fish people, because you can get the egg. It's even easier. You can get the fertilized egg through manipulation, so that it doesn't harm the mother or the father. So that's also one of the advantages of using the chicken. So again, with these germ cells, we can manipulate the early embryo at high welfare standards, so we don't have to do surgery or reimplant these back into a surrogate host mother. We can do this in the egg. But one of the problems is if you work on these larger animals, so if the chicken is not happy, it's going to happy. So we are very concerned with the welfare of the animals. So, you know, the feather color change, we could observe them to make sure that they were in perfect health. But, and if they weren't, they would definitely have told us if they were, yeah, even in a bad mood. That's the thing they have.
We started out with the academic perspective and the commercial perspective. And the three of you made it very clear that from the academic perspective, understanding development of animals, producing transplants, it is so doable. So I want to emphasize a little bit the commercial aspect. And being a pig breeder, I have 2 dam lines and 1 sire line. And in order to maintain the genetic variation, I have, let's say, 2,000 females per population. So the sheer work to be done to get copies of these animals, and then I have 1 edit or a cluster of edits, but I should repeat it in 5 years' time or whatever. And I might be wrong, that there might be shortcuts or something. Could the three of you speculate a little bit on it? And Mike understands my question, so maybe you would like
I'll say it first because I'm really interested to see what Anna has to say for Björn. I know the problems that they have, but exactly. So poultry is, when I showed you what was really nice, when I showed you the feather color trait that we edited 1 male PGC line and 1 female PGC line. So what was really nice, I mean, what was interesting is everyone we produced was a huge flock of full sibs, but they were closely related. Now, if you would want to do this for a production trait, well, for poultry, the flocks contain hundreds or thousands of genotypes, you know, of different genotypes. They won't want a bottleneck, so they won't want to go through 1 single edit. So we haven't even thought how we would model that to be able to edit with diversity. It's such a hard question, especially for poultry. Yeah. Okay. Maybe the other two. Yeah.
Oh, sorry. Anna, you have a challenge?
Yeah, I can say something there. I mean, what we see, of course, a very big mosaicism, but we see some common mutations, so we can sort of cross F0 to F0. But what we also see is that it can be very useful to use HGR. So we use HGR to create a very high degree of 1 type of mutation. And then we can do that, I guess, in several nucleuses. We have discussed with the breeding company. But the thing is, very, very big investment for the breeding company, because they have to make a parallel breeding company, you know. So they have to at least take out. They have several nucleuses and introduce it to all of them, I guess. So that's very, very interesting. And I think, but when you have a very interesting genotype, the investment is worth it. So I think that's when it's coming into use.
Okay, thank you. Mike, I interrupted you twice because of the timing thing.
No, fine. That's fine.
Okay. Björn, you want to reflect on that?
I agree totally with the pig. I mean, this is really hard to estimate how you can bring it into a commercial pig breed, but I think it's important on the gain that you have from the genetic modification. I mean, you can achieve it, and maybe, and we're now, they're also working on new delivery methods. I mean, if you maybe could add the CRISPR to the sperm that you sell to the farmers, maybe, and then they do the edit in their piglets, maybe. So that would be maybe another thing, but it's important for other livestock species, like in cattle, for example. It's much easier to propagate specific traits in these species than in pigs.
Thank you. And Kasia, did you find new questions? And if not, I know that you're interested in the cell and organoid mutations, and then the translation to animals.
Yes, thank you, Egbert. No questions at this moment from the audience, but another for the speakers. If you could elaborate your opinion, are cell and organoid results that you obtain today in gene editing representative for the live animal?
It depends on the edit.
Wish to take that.
It depends a little bit on the edit. I mean, how complicated the thing is. For example, with the African swine fever, I mean, we had some differences in the in vitro results where you just have the cell type and then you have the virus. When you then generate a complete animal, I mean, there's some other factors that play a huge role, the environment, the immune system, and all that stuff. So it's really hard to predict results that you achieve in the in vitro assays if they are the same in the offspring afterwards. But I think some easy targets where you already know a lot about and there are not so many factors involved, I mean, then you can also use it in organoids or cells. But I think it really depends on the target.
Maybe I'll go next. Another problem with avian species is that we don't have a lot of cell lines. We have a few cell lines, and we don't have many organoid cultures. So we're desperately trying to catch up. We have Lonica Verveldi, who has her lab here, who can grow chicken intestinoids or enteroids. So we're finding that a really good system. And the idea is anytime, now that we have the technology to edit chicken, pretty successfully, the first thing we want to do is not to edit the chicken. We want to be able to screen in cell culture before you make the animal, because it takes too much time. It's 5 months per generation, and it's too expensive. So now it's like any work we can do in cell culture is well worth it. Even for any models we have, we see that as the way forward.
Yes, thank you. Yes. That's true. That's what I also believe, and that's why we also try to do it. Yes. So I think, Egbert, that we are actually run out of time now. But even if we wish to keep going, then our webinar clock is over. But we really thank you all for your contribution. It was really exciting to see you, to hear to you. And Egbert, do you wish to add something?
No, that's beautiful. I'm just wondering if Andrea Rosati, as a representative of the EAAP, would like to have the final word in this session.
And invitation to that next one, yes?
Well, thank you, Egbert. This was a kind of tricky invitation because I was not expecting that, but I'm here, so I'm happy, and thank you very much for giving this opportunity. First of all, I want to thank, I think Katarzyna was doing the same. I mean, to thank all the speakers who presented a very interesting topic. I enjoyed very much. I learned a lot, and I guess I'm sure that the rest of the audience, they share the same feeling, the same impression that I had. So thanks very much for your presentation. They were extremely interesting, and as well as thanks to Egbert and Katarzyna to have organized and selected the speakers as such good speakers and a very good topic. So thanks to all of you. It's certainly important for the Animal Science Network in Europe and beyond Europe, of course, to learn new techniques as the ones you discussed today. Having said this, I have to look further to the future, and on the 23rd of November, so a little more than 1 month, we have the number 7 webinar of EAAP that is open to all interested audience. It's free, it's open to everyone, and will be about the genetic again. And as Egbert announced it a little bit in the beginning, it will be kind of like a launch and for the World Conference of Genetics and Applied Livestock Production that will be held in Rotterdam early July 2022. So this webinar will be on genetic topics and announcing the next World Conference on Genetics. So thanks very much. See you again soon here at the EAAP webinars. And again, I wish to be there to clap all the speakers and Egbert and Katarzyna. Thanks again. Bye-bye.